So modern medicine, I guess, why is modern medicine important from talking about normal traditional medicine, right? So anything that is modern, right? Any of the medicines that you see right now has come after so many years of research, so many years of development. So it's important that we talk about modern medicine. We talk about how medicine has evolved, and we'll get to this two-part session, right? We'll get to what the most latest technology is, I think, as we move progress and further on. But now we'll start to be able to understand modern medicine. We'll slowly evolve into modern medicine and start from the beginning, right? Let's see, how did we get there? How does it make sense? And what sort of input and response are we looking for? So as he mentioned, right, this is going to be an interactive session. So I have questions. I don't know, I might not know answers to these questions. So I'm going to ask you guys these questions, and you'll have to tell me, okay, some of these answers. So the first question, very simple. Just want to know from your point of view, right? So have any of you taken any medicines before? Anybody, any of you or anyone in your family or anything like that, have you guys taken any medicines? If you have, just tell me a name, one name, two name. I just want to know out of pure curiosity. Have you ever felt sick? Have you ever had to take a medicine? If yes, what is the name? What have you taken? What medicine have you taken?
Panadol.
Okay, awesome. Yes, that's a common one. Yes.
Paracetamol for fever.
Correct. Paracetamol. Yes, that is the most common one. Any more? Paracetamol. Yeah. Yes, definitely. So very simple, right? Those are some of the few most common ones, right? Paracetamol, neomycin, someone has said for ear infection. That's very accurate, right? So as a quick example, yes, I've just given an example, right? Crocin, right? People take crocin for fever. People take Benadryl for cough, right? Gelusil for other things. These are the most common ones that there are. So now I have a question too. What is in crocin or what is in Benadryl? By that, I mean, what are the ingredients? So for example, let's take an example of Lay's, right? Lay's is what? Potato chips, right? So if you take a Lay's packet and you look at the back, it has these ingredients listed in it, right? So there's potato chips and oil. Those are the two things that you need to make chips, right? You need to cut the potatoes into pieces, fry them in oil. What is everything else? Everything else also, if you look, it's sugar, salt, onion powder, spices, all that stuff, right? You need to make sure the potato chips taste nice, right? So when you look at this set of ingredients, all right, this makes sense, right? This is, everything that you need to make a medicine. But have you, like all the things that you mentioned, right? Neomycin, paracetamol. Have you ever looked at the ingredients at the back? Do you know what is in there?
So some nutrients like carbohydrates and then starch, and then a composition of medical drugs and maize.
So I asked if there's any specific names of these chemicals that you just mentioned, right? You said mixture of therapeutic drugs. You said, so tell me, what do you have any specific names for these drugs that you remember in the back ingredient list?
Sir, I'm not sure if this is for Panadol, but I guess it is aspirin. I've frequently heard aspirin.
Yes, aspirin is definitely a common chemical compound. Yes, correct. Absolutely. Anybody else remember any names that you might have seen? Anything that you might have seen in the back label of these pills? Okay, so just as an example, right? So I showed you Lay's, right? So this is what Lay's looks like. Easy to understand, right? It has potatoes, oil, all these seasonings, all these flavors, so on and so forth. But this is an example of something called Tylenol, right? It is used for fever, right? It is like paracetamol and like crocin and so on and so forth. So if you look here and you see one active ingredient, right? One active ingredient on the top highlighted in yellow, that says acetaminophen, right? So that is specifically the actual drug that is in the pill to help you feel better. So if you look at Tylenol or crocin or anything like that, you will see this. You will see acetaminophen. You will see its name because that is what we always use, most commonly use. to be able to make our cells feel better. So you'll see that that is the active ingredient. But if you scroll here, you'll see inactive ingredient. All right. So what is an inactive ingredient? If you take a look, you'll see that it's okay. It's wax, starch, but it's a whole bunch of very complicated names. Aluminum lake, hypromellose, magnesium stearate, polyethylene glycol. So if I just told you, right, I just told you that that there is the active ingredient, which is the acetaminophen, right, which is what makes you feel better. Well, if this is the active ingredient, why do we need inactive ingredients? What is the point of having an ingredient that is not active necessarily?
So
Anybody
to
has any
give the
guesses?
solid structure for the specific medicine.
Absolutely correct. Perfect. Yes, that is definitely one of the biggest reasons. Yes. Any other guesses? Any other reasons why you may think that we might need so many things? All these complicated looking chemical names? Easy, right? Why do we need all these complicated ones? Like you said, absolutely correct. Some of them. So it's easier to always eat a pill, right? It's always easier to eat a medicine that's in the shape of a hard pill. It's much easier to do. So some of them, it might be in powder before, like a chemical, like acetaminophen might be a chemical powder. And you might use some of these things to make sure that they become like a solid pill that you can, tablet that you can eat. So any other guesses for maybe other things? Why we might need all these inactive ingredients?
Taking the drugs to the proper place to deliver the drugs to the body.
Okay, yes, some of them might be needed. Yes, that's also very good. So as you correctly mentioned, right? The first one of the biggest reasons is to give it a structure, right? So when you make a chemical, it might be a powder, sometimes it might be a liquid, but it's not always the easiest, right? So you don't want to eat a powder; it's very difficult, right? But it's easier to eat a tablet. It's easier to eat a small pill, right? So some of these might be required to give it shape, a good, nice shape. What you said about going into the right part of the body, yes, some of it is. So for example, let's say that there are specific pills that are made so that, let's say, if it is meant for a stomach disease, then it only breaks and dissolves when it comes to a stomach. So the outer covering is such that they have enough chemicals such that it doesn't dissolve when it goes in your throat, it doesn't dissolve when it's in your throat, it doesn't dissolve when it's in your drugs, it only dissolves when it's in your stomach. So yeah, some of that is definitely the reason as well. But a lot of reasons why we have inactive ingredients mainly is also because, well, to make it stable, right? You order a pill, right? And then it sits outside on a shop for 6 months, and you can buy it 3 or 4 months later, and it is still active, right? It is still fine. So for things like, to make sure that it doesn't, you don't have to freeze it all the time, or you don't have to put it in the fridge all the time. Some things might make it more stable. So things like inactive ingredients just make it better, just make it safer to eat even after 1, 2, 3 weeks. So a lot of reasons why, correctly mentioned, solid order is one of them, stability is some of them, and to make sure that they have the right ratios, right? For example, acetaminophen might be very, very helpful, but they might have some side effects. So to counter the side effects, we might add some of the other things here, right? To make sure that it doesn't cause any harm to anybody else here. So this is obviously, right, the newest medicine form, right? Tylenol or any of these that you mentioned, crocin, paracetamol, neomycin. All these are new medicines, right? But how did we get here in the beginning, right? We didn't come here directly, right? So we've been using different plants and minerals for thousands of years, right? I mean, even if you go back and look back at paintings, then you'll see like the paintings on the right where people are using different medicines, different plants to treat diseases that we've had, right, for thousands and thousands of years. But back then, they didn't necessarily have people working in the lab, people working as scientists to kind of help develop medicines for them, right? So more often than not, just because of the way society was structured, cures and medicines had more to do with religion, right? So you'd go to the temple, or you'd go to the church, or you'd go somewhere else, and then you would ask, that person, hey, can you help me with this? Can I make feel better? So in the olden times, at least, society was very, I guess, medicine was very involved, and religion were very involved with each other. So usually these were folk cures, right? It had more to do with religion than science. But even back long time ago, there have been instances where all these temples and churches have had small labs, small compounding labs, where they used to try to make some versions of these medicines, right? Might not always be accurate, might not always be the best thing, but there were at least some sort of like place where they used to mix chemicals together and they try to make sure that they're making like a medicine that might be able to give to the other people. So I have a question. How do you think these remedies were created? So like I said, right, think of this as thousands and thousands of years ago, right? I am telling you that there were no labs, there were no encyclopedias, there's no research, no internet. How do these remedies come about? For example, I'm sure that all your grandmothers have said that tulsi is good for you, right? When you're a cold, ginger is good for you when you have a cold, right? So how did you think they know, or how did you think they found out that tulsi and ginger maybe were good for cold and fever and so on? Any guesses on how that might have happened?
By testing it.
Sorry, can you repeat what you said, Ritikesh? I didn't hear you. Sorry.
By testing it.
Okay. Yes, testing it.
On a
Okay.
patient who is cold or fever.
Okay. Yes, absolutely. Correct. Any more? So yes, right. So the idea would be that they were testing it, right? They had some people who ate it, right? For example, maybe my neighbor ate ginger when he had a cold, and he said, "Oh, I feel better now, right? I feel better, so I'm going to also tell my other neighbor that when he has a cold, he should eat ginger, right?" So this is very good, right? People speaking to each other, people relying on other people for information, which is always useful, right? But sometimes they're not always accurate, right? Let's just say that I ate ginger and I felt better about my cold or my fever, but you might not feel better about my cold or my fever, right? You might not feel better with ginger. Something else might work for you, like tulsi might work for you, and some of them might be poisonous. For example, if you like ginger and it works for you, that is perfectly fine, right? But I might be allergic to ginger, so what happens then? So the same person who prescribed ginger to me and you, well, it worked for you, but it did not work for me. And while that is definitely a useful medicine, it is not ideally the perfect way, right? And some of them might have been even poisonous, right? There have been people who said, eat this leaf, eat the other leaf. Well, what if you pick the other leaf by mistake? There are fruits and there are leaves that turn poisonous after 1 week or after 2 weeks. So if you ate it before that 1 week, it is perfectly fine. But if you ate it after that 1 week, it's poisonous now and you die, right? So these are all dangerous techniques. But some of them clearly did work, right? One of the most common things that we know from before is that there was multiple examples how all these things have gone in, right? So now you see a lot of pills, right, that have tulsi essence in them, that have ginger essence in them. Some of them worked, and I think the most common example is that there is this plant called milkweed. I forgot what it's called in Malayalam, but and usually it has been used for a long time for traditional treatment for like tumors. And now, after years of research, they were actually able to make an effective drug out of this. There's actually a drug called Picato. I think it is separate. It is manufactured by this company called Leo, and this drug was actually created based off of ancient tradition and history, from talking about what we get from milkweed necessarily. So the old way of method of drug development and making medicine were useful, has always been useful, but it is not, I guess, necessarily a scientific way. The idea is that some things might work for some people. We might not have enough information altogether to be able to manufacture this drug completely.
Liquid
Maybe I എനിക്ക് actually പേര് അറിയില്ല. Maybe you might be right. I'm not sure. So as I said, in the beginning, medicines were all about religion, and we might not necessarily find a better way. So the first person who actually maybe stepped away from it and started making medicine in the lab was this Austrian pharmacist. His name was Frederick Serturner, right? He was Austrian. He lived in Germany for the longest time. So what he noticed was that you've seen opium, right? Opium plants. So that anybody who eats opium feels sleepy. So he wanted to know, well, what is the reason? What is the reason for that? So he slowly and steadily started boiling opium in his house, in his lab, and then he started isolating some of those ingredients from opium, and then he fed it to different people. He fed to stray dogs, he fed to some of his friends, and then he realized that they could get very good sleep if he isolated some particular compounds from opium. And they took this and he said, well, this is the first medicine. I called it morphine because the Greek god of sleep is called Morpheus. So he named it after Morpheus and called it the morphine medicine. Like I said, he tested his drug on stray dogs and on 3 of his friends, right? Now imagine, it worked out okay, but imagine if it was a poisonous drug, right? He could have killed the stray dog, he could have killed his friends, right? There is no regulation, there are no rules back then. So essentially, this was just him trying to see, out of his own curiosity, trying to make a medicine because he noticed that he observed that anybody who ate opium was feeling sleepy. He inferred that there might be something in opium that might be making people sleepy. And he said, well, you don't need to take opium, eat opium to feel sleepy. If someone has any trouble sleeping, maybe we can just take that one thing from opium that helps people sleep, and then maybe we can inject it or give it to them in an IV bag or something like that. So this first process, he was essentially the first person to actually create a medicine in the lab, and this was what, 1804, right? So that was 200 or so years ago. So of course, now you can't test any drug on dogs or your friends. You have to go through a very formal process, right? And we'll talk about this. But in 200 or so years, there have been so many developments, so many new diseases that we've cured, right? Imagine that all these issues, for example, you said you had a fever and you took crocin, right, or paracetamol. 200 years ago, if you had a fever, you might have died, right? If you had a headache, you might not have gone to school for 3 days, and you might not have been able to write exams. So the fact that in 200 years we've made so many progress and invented and discovered so many medicines to help make your life better is just phenomenal, right? So to start it off by him, right, in 1804, he made a sleep medicine, and this I'm sure helped a lot of people at that time. And then a company took over this formula, this process, and this was when the first pharmaceutical company started. They in fact made a better process, right, in the lab for manufacturing morphine from opium, and then they marketed it and they start selling it to patients. So, like I said, from starting from sleep medicine to fever, now to cancers, most recently COVID, right? We've been using science for so long, right, for 200 years, and we have been getting better, right? We've been getting better with the year to make sure that, we're developing better medicines every year we go through. And each drug that you see, right? You see paracetamol, you see neomycin, you see Benadryl. It comes after multiple years of effort, right? So many years. There have been people in labs and companies trying to figure it out, trying to make sure that they are developing a medicine that is safe and that is careful and that is very effective, right? You don't want to eat a medicine that the doctor says and say, I still don't feel better. There's no point in that, right? So there are lots of people who spend a lot of time trying to create these better drugs. So how does this happen, right? How does drug development happen in a company, right? What are the steps? Well, you might have heard, right? So it's research, it's development, it's manufacture, and it's post-approval maintenance. So let's start. What is research? What necessarily happens here, right? So in research, well, we talk about what are the diseases, right? First question is, what disease are we looking to cure, right? And I'm going to take the example, let's say cancer, right? We're trying to cure cancer. All right, so what cancer are we trying to cure? Are we trying to cure lung cancer? Okay, got it. I'm going to try to cure lung cancer. So now we have to go into studying what is lung cancer, what happens in the body, what in the body causes lung cancer. Is something from outside the body? Is it like a virus or a bacteria? Or is something that is inside the body that happens that causes cancer? If it causes cancer, lung cancer, where exactly in lung does it cause cancer? And if it does, if you find out that, then good question. Then question is, well, what can we do to help with it? Is there a chemical? Is there a medicine? Can we find it and can we put it in your body, right? So identifying what the disease is, where in the body the disease is, and what medicine we can use. That is the research part of it, right? And lots of years, right? Lots of years of research is needed to help cure a drug and thousands of people, right? So even for something like, 20 years ago, if you had cancer, you were almost 20 or 30 years ago, you're almost sure to die. You didn't have a disease, you didn't have a cure for that. But right now, because of all this, there are, even if you have cancer, there's a good chance that you will be okay, right? You will be perfectly fine because we have enough medicines right now. So that is the research part of it, identifying what is the cause for the disease, right? What is the disease and what can we do or what is needed to avoid or cure the disease, right? So this would be a part of research that would be answering the question. The next part is development, right? So now you're saying that, wait, I know what the disease is. I know where in the body the disease is. I know what we need to cure it, right? So the next question is development. What can we make something? Can we make a chemical? Like I said, can we make a crocin? Can we make a neomycin? Can we make it? So the answer might not be possible, right? So you might know the right drug or the right target or anything like that, but you might not be able to develop something. So again, years goes into making the right pill, making the right, syrup, making sure that the syrup is safe, making sure that the pill is safe, making sure that once all of this is done, that you're still able to deliver it to the patient who's feeling sick. All right. So that's why it's very important that development focuses on something that is effective, which basically means that when you eat it, that you feel better. When you drink the syrup, you feel better, right? Effective. It does cure your disease and safe, right? So I'm sure that anybody who's taken Benadryl has said, oh no, I'm feeling sleepy, right? So that is a side effect and that's fine, right? You have a side effect from eating a medicine, sleepy. It's not the worst problem. But if your side effect was that, that your nose is bleeding or something like that, that's not good, right? So development focuses on making sure that what drug that you're trying to make is effective, that is, it helps you cure the disease and is safe, and that you're not going to fall sick because of the drug. The third step is once you've done the research, right? Once you've developed it, right? You have something in the lab that you think, okay, now I think that we can use this medicine to cure a disease, right? So now you focus on manufacturing it, right? So what is the key thing in manufacturing? Anybody can build a large factory, right? Anybody can buy a large factory with equipment and instruments and say, you know what, let's put whatever reaction that I did in a lab, right? You have done reactions in school, right? I'm sure all of you have done reactions in small flasks, in different vials. And if I tell you that, I'll give you a bigger reaction or a bigger flask, I'm sure that a factory is not that difficult to do. But what we need to make sure is that that drug, whenever you manufacture it, is the same all the time, right? If you manufactured on January 1st, 2021, there has to be the exact same thing in January 1st, 2051. Because if you take crocin now and if you take crocin 30 years later, it can't be different things, right? It has to be identical. Like, remember I mentioned all those inactive ingredients and the active ingredients? So many complicated chemicals, right? Now, even if one of them is different, then it might not work the right way, right? It might not cure the disease that you have. It might be more poisonous, it might be less poisonous. You don't want any of that, right? You want it to be exactly the same. So you have to control the process of manufacturing so well that even if it's 30, 40, 50 years down the line, you're making the identical drug substance every single time. And the last is post-approval maintenance. This isn't necessarily complicated. Once it goes into the market, that is once that you release it into the to the people, you make sure that you're monitoring them, right? You make sure that the patients are continuing to benefit from the drug. So that is why if you look at any medicine, there'll always usually be a number, a phone number that you can call and say, oh no, I've had some side effects, or oh no, I'm not feeling as good. You're supposed to call that and inform them, right? So they know that, for example, if something, if they manufacture something, sold it to the store, but it is not working well, they know it's not working well, right? And then that is a part of, and they have to go back and say, wait a minute, is this an issue with the manufacturer, or is this an issue with the development, or is it an issue with the research? So they have to make sure that they have to continue maintaining it, so that they know, even if after they develop and research and manufacture, even 30, 40 years down the line, they know what is actually the issue and if there is an issue at all. So I just told you, right? Research, development, manufacture, post-approval maintenance. And we also spoke about, how maybe 200, 300,000 years ago, maybe the way in which we knew about medicines were about talking to each other, things like that. So now that I told you the scientific process, do you think the scientific process is better than the older way of developing medicines, or just talking to each other, or trying to go to a religious place to get medicines? What do you think is better? Which process do you think is better?
So the modern medicines.
Why do you think it's better? What is better about the modern medicines part of it?
Sir, it is more effective.
Okay. Correct, right? Absolutely, right? Because we're building in so many steps, right? We're focusing on making sure that each step that we do, that there is a reason why we're doing it, right? Research, development, manufacture, right? We're controlling the process, making sure that that everything that we do is accurate. Any other comments based off of the last slide? Do you any more suggestions that you think might be good for the scientific process?
Because the people who are making it have the required amount of knowledge what to make and how to make.
Absolutely. Every single person who's involved in that process, right, that 4-step process, has gone through multiple years of studying, right, multiple years of work experience. They're also paid quite well. So they're happy to do the job, right? They know how to do it. They have multiple degrees, bachelors, masters, PhDs, right? Postdocs, years of experience. So yes, absolutely. These are professionals who know what they're doing. Yes, correct. Fantastic. So I guess one more question here, right? So I just told you, right, the full process: research, development, manufacture, post-maintenance, right? So let's say I have something in the lab, right? I made something in the lab. I think it is going to be very, very useful. I think it is going to be able to cure a disease. Okay. What do I do now? Should I say here? Should I start giving it to people and saying, here, this is a medicine I made in the lab. Can you please take it? It's very effective according to me. I did experiments in the lab. Okay. I did experiments. I think it's 100% effective, 100% safe. Here, take it. Should that be the way it works?
No, sir. I think we want to test first for few people, very few people.
Okay, perfect. Yes, that's a good suggestion. Anybody else have any ideas?
Yes, sir.
So we can't, because we need to go through multiple tests.
Mm-hmm.
And then we also need to have a license to sell our drugs.
Yes, again, perfect answers, right? Perfect answers. So like you said, testing, right? What sort of testing can we do? How do we make sure that what is in the lab works, right? So throughout the time of research, right? Throughout the time of research, to the time of development, we're always doing testing, right? What are we doing testing on? Well, we're doing testing on mouse, right? We're doing testing on mice. Well, everything that you make, you see, wait a minute, So we made this drug, we made this research, right? How do we know it is active? Well, the first step is, while doing some experiment, you try to see, well, does it work on mice? If this mice had fever, can we cure the mice of fever? If the answer is no, then maybe it won't work for humans, right? Why would we think that if it doesn't work for mice, it would work for humans? Why mice specifically? Because of a number of reasons. The physiology and the way the body works of mice is quite similar to humans, not completely similar, not very similar, but it is more similar to humans than most other animals, at least. Or not most other animals, but definitely something that is easier to control. Like dolphins also have something very similar, but it's very difficult to test on dolphins, right? Imagine having an aquarium trying to figure out how dolphins, right? Mice are in cages, right? Very easy, right? You can have 100 mice in one room, right? And then you can do an experiment on 10 of them, see if they feel better, right? Hopefully they feel better. And then you progress on, right? So you have, during research, you say, here, I have shown you that this works in mice. Now what do we go to the next stage? Well, monkeys, right? Why monkeys? Because they're the most similar to human beings, right? This is as close as we can get without testing in human beings. Now, why do we need to do these tests, right? Well, like I said, everything is a complicated process, right? This is a complex process of making chemical drugs, right? How do we know it's safe, right? So these first two steps we're trying to answer, is it safe? Can we give it to mice? Can we give it to monkeys? Are they okay after that? Because if they're okay after that, then we can move on to exactly what I said, is testing it in humans. Now, this is also a process, right? You can't just give it to 10 people and say, please test. It has to be a very controlled process, right? And I'm sure you've heard of this process. It's called clinical trials, right? So clinical trials usually happen in a very controlled manner. So this is how it works. There are 3 phases, right? Phase 1, phase 2, phase 3. And each of these phases have their own requirements, have their own needs, and they're designed in very specific ways. So phase 1 is, it's done with a smaller group of people. Okay, it's not done with a large group of people. It is very limited amounts, depending on the disease, depending on the reason. Maybe 25 people, maybe 50 people, maybe 100 people maximum, right? And these are not given to people with the disease. Let's say it's a lung cancer drug, right? In the first phase, you don't give it to lung cancer patients. Why do you not give it to lung cancer patients? Well, the first phase is only purely to know if your drug is safe. That means if someone takes it, are any issues caused due to the drug itself? And that's not good, right? If I gave you crocin to cure your fever, but if the crocin itself is poisonous to you, then that's not good, right? It might cure your fever, but it is poisonous. It doesn't help, right? So the main reason for doing phase 1 trials is to make sure that you know if the drug is effective. And if you do that with someone who's already sick, you might not know, right? For example, if you're giving a lung cancer drug to a lung cancer patient, and if they unfortunately die, you don't know if they died due to lung cancer, or you don't know if you died due to the drug because your drug was poisonous, right? So you want to avoid confusion. You want to make sure you have very clear data, make sure you have clear reason as to why, and if your drug is poisonous or not. So it's given to a small group of people, okay? It's given to healthy people, right? People with no disease, right? And to make sure that and they take it, and then do they have any side effects or not? Now, it sounds a little dangerous, but it's not, right? Because we have data from mice and humans. So we have a rough estimate, right, already to see how much will be poisonous. So let's say that if, for example, if it was found poisonous, that 1 milligram of the medicine was poisonous in monkeys, then 1 milligram will also maybe be poisonous in humans, right? So you will never give anyone in these here 1 milligram. You will give them maximum. You will say, I'll give you half a milligram. Okay. And then we'll see if half a milligram cures you. If it comes to a point where we say that, half a milligram is not poisonous, but it can cure you, that it's not, that we have seen in the mice and the monkeys that half a milligram can actually help, then you go to phase 2. At phase 2, again, now you do it with a larger group of people, right? Rather than 25 or 50, now you do 200, 250 people. Now this is with people with disease, right? So you already know it's safe, right? You already know that it is thing. Now you go here. Now it's a lung cancer drug. Now what do? You give people, 200 people, 250 people this medicine, right? And then you see, well, how do they feel? How do they feel after 2 months, 3 months, 4 months? Right? And you measure, do they have a tumor in their lungs? And then you measure the size of the tumor, right? Every time that they pass through the month. So 1 month, you look at the size of the tumor. 2 months, you look at the size of the tumor. 3 months, you look at the size of the tumor. If it decreases, then you know, wait a minute, I think our drug is working. And you also look at safety, right? You're also, even with the larger group of people, you're making sure that no one else is falling sick, you don't see any side effects, so on and so forth, right? So phase 2 is important because you're now looking at effectiveness to seeing, yes, can I cure the disease? Is my drug actually working? And phase 3 is the last phase, right? It's on the larger group of people, but also more diverse, right? So for example, if this drug is being made in the United States, maybe phase 1 and phase 2 will be completely done in the United States, but phase 3 will be done all over the world, right? Because essentially speaking, there should be no differences between someone who has eats the same drug in the United States or India or China or Russia, right? But there are sometimes differences, right? So it is done with a larger group and a diverse group of people. And here again, you're making absolutely sure that your drug works, that it is effective. And another thing here that you look at in phase 3, is it better than what is already in the market, right? Let's say that there is crocin, and crocin helps you cure your fever, right? And you're making a drug. Well, it cures your fever, but it doesn't cure it better than crocin. Then what is the point, right? What is the point of a new medicine? There is almost no point. You can maybe demonstrate saying that, you know, this is cheaper, this is, you know, crocin's 100 rupees, I'll sell it for 50 rupees. Then maybe you can say that, you know, not as effective, but as a cheaper drug, it still helps cure you. It's not poisonous. So we will still go ahead with it. So phase 3 is important. It's the last step of phase 3. So it's obviously, right, very long, very complicated process. And this happens in the same timeline as research and development, right? So phase 1 can only start after development and manufacture, right? You need something to be able to do that. So while phase 1 and phase 2 is going on, you still continue to do development. You still continue to make sure that you have a better product. So what is the level of success in phase 1 clinical trials? So phase 1 clinical trials, it's 75% success rate, right? So 75% of the medicines or the drugs that these companies, these labs, these people put into phase 1 clinical trials, 75% success. So which basically means that, you know, from the process of, you know, animal testing and, you know, mice testing and monkey testing, they already know 75% of them are already successful at making a safe product. Now, when you go into phase 2, only 33% of it is successful, okay? Why? Well, you know, your product might be safe, but it's not curing the disease. Okay. And phase 3 is even more harder. It's 25% success rate. So think about 75, 33, and 25%. If you do the math, right, very quickly, if 100% of the drugs go into clinical trials, 75% succeed. Okay, so 25% are gone here. Out of that 75%, 33% succeed, right? And out of that 33%, 25% succeed in phase 3. So which basically means that of 100% of the drugs that go into clinical trials, 6% succeed. Okay, so if 100 companies try to do clinical trials on one drug each, only 6 of them succeed. Now, how much time does this take? This takes about 6 to 8 years, standard. On average, it takes about 6 to 8 years for a complicated cancer drug. And it takes 1.3 billion US dollars for each drug for research, development, and clinical trials. Why? Any guesses on why it needs so much time and so much money? And why is there only 6%, right? I mean, 6% seems to be a pretty low success rate for anything, right? If you told someone in your chemistry lab, right? You went to the chemistry lab and say, you know, I'll do this experiment 100 times, but only 6 will succeed. No one's going to be happy, right? Your chemistry teacher is not going to be happy. Your parents are not going to be happy, right? No one's going to be happy, right? So why? These are all companies with so much money, right? Why do they take so much time? Why do they need so much money? And why do they still fail most of the time? What do you guys think? Any answer is the correct answer. So go ahead. Guess. I don't have the right answer either, right? Complete process. So guess why is it going wrong? Why might it be going wrong?
Can you repeat your question?
So like I said, right? So clinical trials in humans, like I said, 75%, 33%, 25% success rate. So that means that on the whole, only 6% of it is successful, right? So 95% of it fails in clinical trials. And it takes 6 to 8 years and it takes $1.3 billion. I guess my question is, if you're spending so much money, if you're spending so much time, why is it still failing? Why is 94% of clinical trials still failing? Like, why is it so difficult? Like you said, right? The people, the experts are the ones, right? They are professionals doing the work. We've already done animal testing. Why is it still failing? Why do you think we still might be failing in clinical trials? Like, why are they not able to, 6%? Why are they only succeeding 6% of the time? Why are they failing 94% of the time?
Sir, I didn't understand the question.
So let's say that I'm trying to treat a medicine, okay? And I make 100 medicines, but only 6 of them are successful, or 6 of them are good, and 94 of them are bad. Is that good or is that bad? It's not good, right? Because 6 out of 94 is too small of a rate, right? If you say you're good at something, then you at least have to be 50%, right? If you get 6 out of 100 marks in an exam, is that a good performance? No, right? Because it's not. Yeah, correct. Absolutely, it's not good. So we have to be. So that's my question, right? My question is, why is it so low? I think I saw someone on the chat, right? It says it'll create different effects in different people. Spot on, right? Very accurate, very correct, right? And that is why we do this complex process, right? That we want to make sure that it is having the same effect on everybody, right? If it's having different effect on different people, then basically that is saying that your drug isn't effective enough, that it's not good enough to be marketed. Any other reasons why it might be failing, or it might be not be succeeding as much? Guesses?
So safety issues?
Absolutely correct. Absolutely right. So safety issues is obviously very important, right? Because not all of them are we able to create as like, but also very simply, right? It's just very complicated, right? It's just very complicated. So Yisheng said that I saw Nandapriya's message say that done with a smaller group of people. Yes, but phase 2 and phase 3 are usually done with a larger group of people, so it's usually fine. And yes, safety is definitely one of the biggest reasons why it fails. Anything else? So one of the biggest reasons it fails, right? It's still very complicated, right? We still, when it's a complex disease like cancer, right? It's still very complicated. Like I said, research and development 6 to 8 years, right? Like we still don't completely understand our human body well enough. We still don't understand disease mechanisms well enough, right? We're still trying to learn. We're still trying to know more about ourselves. So at this point of time, our effectiveness is low because we just don't know enough. We just don't know. We just don't know why it isn't working. I see Isabella's comment where they say that maybe it's because of the regions where they live. Also, accurate. Unknown side effects. Yes, absolutely. These are all usually the reasons why they do fail. Usually because in the regions, right? For example, phase 1 and phase 2 might be done in the United States, but phase 3 might be done in other parts of the country in a different region, and it might not work as well, right? Sometimes we don't know why. Sometimes we might not have a better understanding, but other times it might be easier, right? Like unknown side effects, like poisoning, like safety is not good enough, so it's easier to answer. But overall, this is just a very long and complex process, right? So the difficulty, right? The difficulty is the difficulty, right? So we need smarter people, right? People like you, right? People like you to study, to become smarter, to become better than all of us, so that you can do this better, right? Maybe when you guys start working, it'll be 100% success rate in phase 1, 75 and 50, right? So it's an improvement. So now I just said, wait, this took 6 to 8 years. So on and so forth. But the COVID-19 vaccine, it came out in 2 years, right? And it is pretty successful, right? 100% success rate, right? Almost 99% or success rate. Why did that take only 2 years? What do you think? What happened different there? What change made that 2 years and not 6 years, maybe like usual? Anybody has answers? Why you think that it might have been 2 years? That happened very quickly, right? It didn't take all the research time. Why?
A lot of companies aim to make drugs at the same time.
Correct. Absolutely. Because the entire world was working on it. Correct. Yes.
Sir, please tell one more time the question.
So I said that usually to make medicine takes about 6 to 8 years. But when everyone was making vaccinations for the COVID-19, they were able to do it much quicker, that they did it in 2 years. Why do you think that the COVID-19 vaccine was developed in 2 years and why everything else usually takes longer?
So because this pandemic was a global emergency.
Correct. But what do they do in the global emergency? Like, the importance is enough. Yes, all the professionals came together. Everybody was working on it, right? All these are perfect. So what they usually do is, right? First, any company that is developing the drug, right? First, they do research. So 2 years, they do research. Okay. 2 years, they do development, right? And 2 years, they build and work on manufacturing. Why do they do it? Well, if something fails here, why would they do this, right? So if something fails in research, let's say they're trying to cure lung cancer, right? But they can't figure it out. They've spent so much money, so much time, but they can't figure it out. So they will not do development, right? They won't do manufacture. Why? It is they haven't been able to figure this part out yet. So usually they will do this research, right? 2 years. They will do development, right? Another 2 years. Then they will do manufacture, 2 years, right? Takes a lot of time because they don't want to waste any money. But in this case, what happened was that the government said, you know what, we'll give you the money. So do all these three things at the same time. Do research, do development, do manufacture at the same time. Okay. So when you have ready, we are ready to execute at the same time and cut the losses. Why are companies not going to do this generally on a usual time frame? Why? Because they don't want to lose money, right? Development takes a lot of money. Manufacture takes a lot of money, right? But if the government is saying, here, we'll give you the money. So that even if it fails, no problem, right? You're trying, right? That is more important, right? That even if it fails, that's fine. We'll give you the money for development and manufacture at the same time. So that's how, rather than doing this individual steps, we did this all in one step at the same time, parallelly. And that's how they were able to cut down the time for the COVID-19 vaccine. Any questions here?
No, sir.
All right. So almost all medicines and everything that we've spoken about, actually, everything until the vaccine, everything, all paracetamol, neomycin, everything are chemicals. They are all formed by performing complex chemical reactions. Okay. Something that looks like this. You don't have to mug up this reaction. It doesn't matter, right? Not going to ask you a question on, how the protonation happens or how the cyclization reaction happens, right? It's very important, yes, but maybe not for this. This is a discussion, right? So remember what I said, the active ingredient is acetaminophen, right? So this is a complex reaction, and this is how you get to acetaminophen. So most of them were made in the lab, were made by mixing different chemicals together, by making, chemical compounds, powders, tablets, so on and so forth. So about 100%, all of them, until the mid 20th century, I think about 1960s and 1970s, all the medicines made were chemicals. But slowly and steadily, we started realizing that chemicals might not be able to cure everything. There are so many different types of diseases, right? There are so many different, disease areas. Cold fever is one of them. Cancer is one of them. Genetic disorders, right? One of them. Diseases that are caused due to any maybe environmental conditions, like pollution might cause issues, right? So there's such a wide variety of reasons why diseases occur that we suddenly very quickly realized that chemicals and chemical reactions might not be able to cure all the diseases. Okay. So we had to find a new approach, and that is, now this is where I guess modern medicine starts off at, because we've slowly started steadily moving towards a new technology, and that is where biotechnology has come into play, as where biotechnology and modern medicine now interacted. And rather than making chemicals that cure us, rather than making chemical compounds in the lab, in the facilities, we've started to make therapeutic proteins. So the latest class of medicines, the most effective medicines, have been proteins. What proteins? Well, what have they been able to cure? Well, more complex diseases than we ever even thought of. Genetic diseases, cancer diseases, so on and so forth, something that we weren't able to cure for so many hundreds of years. And before that, when the industry and when all the scientists started making proteins rather than chemicals, that's how we were able to evolve medicine from starting to now modern medicine, right? So I'm not going to go too far. I know we're almost at 1 hour, but I will ask one, last question, I guess, before we stop for today. Okay. What is the first thing when I said proteins, right? When I say proteins to anybody, what is the first thing that comes to your mind? Proteins.
Protein means bodybuilding food now.
Yeah. Absolutely correct. Proteins, bodybuilding, yes.
And it gives us energy.
Yes, it does definitely give us energy. What are the other things that come to your mind for protein? So when we talk about protein, we usually talk about food, right? The moment you say protein, we think of food. Why? Because all these things, right? All the things that we usually eat, all meat, eggs, all milk products, all these, badam, pistachios, they're all protein rich, right? All the seeds, they're all protein rich. So usually when we talk about proteins, we're talking about food, right? But slowly and steadily, right, over the next session, we'll start talking about, yes, proteins are definitely food, but they play a much bigger role in developing medicines. They play a much bigger role in the role they play in the human body and how they can protect us from diseases. And how are the scientists now making proteins that can cure diseases, right? That's a whole new class of medicine, and we'll get to that next session. Okay. Thank you, everybody, for joining today's session, right? I hope that you all learned something. And if you did, I shall see you all next week. And then, until then, have a nice week. And hopefully next week, I hope you guys can learn something as well. Goodbye.
Thank you, sir.
Of
Thank you,
course.
sir.
Of course, no problem.